This is a production of Cornell University.
Oh man, okay, thanks Jenny for that incredible introduction. Yeah, as you apparently all know, you already know my life. My name is Maria, and today I'm going to be sharing some of the work I've been doing for the past couple of years. Can you guys all hear me okay? Okay, awesome. Hopefully by the end of this presentation, I'll be able to convince you that nitrogen immobilization should be included as a weed management tool in an integrative weed management toolbox. So as you all mentioned, I used to work in strawberries, and life was really good because I always had an abundance of fresh and frozen strawberries. And now I work in weeds, and they're amazing, but not quite as delicious. No. So you might wonder why I am doing weed management. Weeds are a really critical component of crop management. So there's a study that looked at crops around the world and saw that weeds cause more crop loss than any other pest management problem. And then there's another study looking at just corn and soybeans in the United States and Canada, and they estimated that without weed control, we could lose up to over 50% of our crops. So weed control is really important for all crop managers, except maybe in controlled environment agriculture. But they're currently facing a lot of issues. So conventional growers rely predominantly on herbicides, but when you apply herbicides to a field, you're exerting a really strong selection pressure on that weed community. And therefore, weeds often evolve resistance through natural genetic mutation to the different herbicides that people use. So here we have a graph showing the increase in herbicide-resistant weeds that we see around the world. And then once a grower has an herbicide-resistant weed community, they often use an additional herbicide on that field. And that exerts a second selection pressure on that weed community. So weeds then evolve resistance to multiple herbicide modes of action. And when I say mode of action, I just mean a way that the herbicide kills a weed. So there are a lot of weed communities that are resistant to multiple herbicide modes of action. And then this leads to increases in costs for the growers. So when a grower has herbicide-resistant weeds on their farm, costs can increase between 50 to 100%. So that comes from increased herbicide applications as well as increased seed costs. But conventional growers aren't the only ones that face weed management challenges. Organic growers could also benefit from additional non-herbicide-based weed management tools. Currently, the options available to them are much more complex, and they also are less reliable and only provide moderate control. And there are often trade-offs between the tools. So one of the predominant methods of weed control for organic growers is tillage, and that can lead to reductions in soil health, erosions through wind and water. So they have to make these trade-offs. So basically, both organic and conventional growers could really benefit from more non-herbicide-based weed management tools. And so that motivated Tony, Jenny, and I to look through the literature and try to find a tool that could be used in agricultural systems, and something that had been used to restore native species growth in prairies over invasive species growth was nitrogen immobilization through carbon amendments. And just so happened that Jed had most recently published in this area, actually looking at landscapes in Hawaii. But we thought we might be able to use nitrogen immobilization as a tool in agricultural systems. But before I continue on talking about nitrogen immobilization, I want to make sure everyone's on the same page about what it actually is. So nitrogen immobilization is predominantly affected or controlled by the soil microbial community. And microbes have a C to N ratio of about 7 to 1. So this is an average. Obviously, there are microbes that have higher or lower C to N ratios, and then microbial communities that have higher, lower C to N ratios. But they generally need a food source of about 23 to 1 because they lose some carbon to respiration as well as other metabolic inefficiencies. But ag soils often have a C to N ratio between 14 to 1 to 20 to 1. So you can see that's a little lower than their metabolic needs. So if you add carbon amendments to the soil, then you can alleviate that carbon constraint. So plant residues really vary in their C to N ratios. So you can have lower C to N ratios, about 20 to 1, and you can have C to N ratios much higher than 100 to 1. So if we choose a plant material that has a higher C to N ratio, we may be able to cause nitrogen immobilization. So basically, if we think about it another way, we're adding carbon to the soil. It stimulates the growth of the soil microbial community. As those microbes grow, they take up nitrogen from the soil, and that leaves less available nitrate in the soil for the plant roots. And you may think that this is going to reduce plant growth for all plant species. And that may be true, but when plants fix carbon from photosynthesis from the sun, they then have to spend that energy either growing or having differential growth. So that may be defense mechanisms such as thorns or metabolites that may help them have reduced disease resistance or increased disease resistance. So because plants allocate their resources differently, they're going to have different life histories or growth habits. So we can see that we would imagine that some plants would grow better in high nitrogen soils and some plants would grow better in lower nitrogen soils. And from the literature and from research, people have noticed general trends that in high nitrogen soils, we often have plants that are better at competing for light. They have higher dispersal ability. They have higher leaf to shoot ratios, and they have higher photosynthetic capacity and may be able to grow faster. But in low nitrogen soils, we have plants that are better at using abiotic resources. They have better or more below ground investment, so higher root to shoot ratios, and they may be a little bit slower growing. So if we put those two ideas together of nitrogen immobilization and different growth strategies in plants, if we have an ag soil where we don't amend the soil, and two plants, one is going to be better at taking up nitrogen, and then it may outcompete the other plant. If we have another soil where we've amended with carbon, we're going to stimulate nitrogen immobilization. There's going to be less available nitrogen, and potentially the other plant is going to be able to be more successful. And many of our agricultural weeds grow really well in high nitrogen soil, so we see a lot of this, and potentially there are crops or areas where we can use this strategy to have more of a scenario that we see on the bottom. So we thought nitrogen immobilization could be used in agricultural fields in some situations. And what I'm going to do from here on out is talk about 3 of my experiments that I did for my PhD. And I'm going to talk about them with specific hypotheses that kind of ran through all of my research. So the first hypothesis was that carbon amendments can enhance nitrogen immobilization. So for that, I'm going to be talking about data from chapter 2. My second hypothesis is that carbon amendments can reduce the competitive advantage of nitrogen responsive weeds. And so for that, I'm going to be talking about data that I have in chapters 3 and 4. And my third hypothesis is that carbon amendments can change microbial community composition and also their function. So I'll actually be showing some data from all 3 of my experiments. So that first question is whether high carbon amendments can actually immobilize nitrogen. Ooh, I just noticed that I have a lot of chats. Is there any questions? I'm on it. Okay, cool. So my chapter 2 data I used to help answer the question of whether nitrogen immobilization can be caused by carbon amendments. And so for this experiment, we had 5-gallon pots that were buried in a field, and we removed all the soil and homogenized it. And then we added a little bit back. We put in this resin bag that you see here, which absorbed anions from the soil. So we could see the availability of nitrate throughout the duration of the experiment. And then we amended half of our soil with sawdust and sugar as high carbon amendments. And then we left the other half unamended and then filled the pots with those different treatment soils. We planted 8 different weed species in the pots, but just 1 species per pot. And we replicated each plant species and soil amendment treatment 5 times. And so we predicted that microbial biomass would be higher and that soil nitrate would be lower in carbon amended soils if we were to have nitrogen immobilization happening. So we looked at this, the microbial community biomass with a chloroform fumigation extraction method. And basically for that, we just had soils and we killed all of our microbes with chloroform and then extracted the carbon and nitrogen from those soils to measure microbial biomass. And then we measured nitrogen availability by extracting the nitrate that had accumulated in that resin bag throughout the experiment, and then using a high throughput colorimetric method to measure nitrate availability. And first, I'm going to show you microbial biomass. We've looked at both microbial biomass carbon as well as microbial biomass nitrogen. And for the rest of my experiment, you're going to see that bars or points that are orange are amended with carbon, and then bars or points that are teal are unamended. So for both microbial biomass carbon and nitrogen, we see a significant decrease in our unamended soils, significantly higher in our carbon amended soils. So this indicates when we're adding carbon to the soil, we are stimulating microbial growth. Then we looked at nitrogen availability with that nitrate from the resin bag. And we see that we had significantly more nitrate available in our unamended soils and less nitrate available in our carbon amended soils. So this indicates to us that we are actually causing nitrogen immobilization with carbon amendments. So yeah, the first hypothesis, carbon amendments can stimulate nitrogen immobilization. And so our second hypothesis was that carbon amendments can then reduce the competitive ability of nitrogen available weeds. So I'm going to talk about data from two different experiments for this. First, we looked at crop weed competition in a greenhouse experiment. And so for this experiment, I had 5 different plants. 3 of them were weeds, and these are common nitrogen responsive weeds in North America, and they're really problematic in many different growing systems. We have Palmer amaranth, Powell amaranth, and then common lamb's quarters. And then we actually had 2 soybean varieties. And then these 2 soybeans had the same parents, but one variety was able to nodulate, and then the other variety was not able to nodulate very well. So we thought that the non-nodulating soybean would respond more similarly to the weeds, and that the soybean that was able to nodulate would be able to rely on nitrogen fixed from the atmosphere and wouldn't be as negatively affected by the carbon amendments and reduce nitrogen availability in the soil. So we planted each of these 5 plants in pots in competition with themselves. So we had 1 pot with 2 plants. We always had 1 pot with 2 plants. So we had monocultures of each of the different plants. And then we also planted all of the different weed species in competition with the 2 different soybean varieties. And then we also had the 2 soybean varieties in competition with one another as well. Half of the pots we left unamended, and then half we amended with carbon. And we replicated each of those pot combinations 6 times. And we predicted that weed above ground growth would be lower, but that soybean growth would remain unchanged in carbon amended soils. And so we used a general linear mix model to analyze this with plant above ground and root biomass as our predictor variable or response variables. And then we had our soil amendment treatment, the plant species or variety in the case of soybeans that we're looking at, plant competition, and then the interactions between soil amendment and that plant species and soil amendment and the plant is in competition with as the predictor variables. And we also included pot as a random effect. So I'm going to show above ground biomass first, and you can see on the x-axis that we have our 5 different plants, our 3 weed species, and then our 2 soybean varieties. And even though you can't actually see carbon amended bars for our 3 weed species, it's because we had really low growth, almost 0 growth in those 3 weed species, and it's significantly lower than the growth of our weed species in unamended soils. And we did not have a statistically significantly lower biomass in our carbon amended soils for our soybeans. So they did not respond, but or at least not as dramatically as our weed species. And then for our root biomass, we see the exact same trend, where we have significant reductions in root biomass for our 3 weed species, but we don't see a significant reduction in our soybean varieties. So that's cool that they are responding differently to the same soil amendment treatment. But we also wanted to look at competitive ability. It can actually be a little bit difficult to measure competitive ability of different plant species. So we came up with a competitive advantage index. And so what this means is that we took the average plant biomass when it was grown in monoculture, and then we subtracted the plant biomass of that same species when it was grown in polyculture. So when it was grown in competition with a different species or variety. And then if the plant biomass remained the same in monoculture and polyculture, then there was no response. But if it was larger in polyculture, then it had competitive advantage. And if it was smaller, then it had a competitive disadvantage. So keeping that equation in mind, for our soybeans, whether we amended the soils with carbon or left them unamended, we had no change in competitive advantage. And it was the same for our non-nodulating soybeans. But our weed species, you can see a significant reduction in competitive advantage when you amend with carbon. So we thought that was really cool and kind of emphasize that carbon amendments can change crop weed competition. We wanted to see if these results would hold in a field experiment. Oftentimes, things that you see in a greenhouse don't translate super well into the field. So we had a field experiment in Freeville where we had 5 different soil amendment treatments, and they increase in carbon as you move to the right, or yeah, to your right. 2 remained unamended. We had a weed-free control and then a weedy control. Then we had a rye hay treatment, a sawdust treatment, and then treatment where we added the sawdust and rye hay together. And so you can see these are increasing in total amount of carbon, but the C to N ratio is actually highest in our sawdust amendment treatment. So this let us kind of tease apart whether total carbon added to the soil or the C to N ratio was more important for nitrogen immobilization and weed control. We planted both corn and soybeans in this field. And the corn we band fertilized to help alleviate some of the stress of nitrogen immobilization for the corn. And soybeans we thought could fix their own nitrogen and again would not be as significantly affected by nitrogen immobilization. We repeated this experiment for 2 years, but the data I'm going to show you is only from the 2nd year of the experiment. We predicted that weed biomass and the community composition of the weeds would vary based on the C to N ratio of the soil amendment. So again, I used a general linear mixed model, and the data I'm going to show you are weed above ground biomass and the community composition of the weeds in the plots. And what we did to get this data is we did biomass clips in mid-August because we predicted that would have the highest total weed biomass throughout the season. We identified all of the weed species within a quarter meter square and dried them and then weighed them. So we knew who was there and also how much was there. Our predictor variables were just our soil amendment treatments, and then we also included block as a random effect. And we analyzed corn and soybeans separately because they were on 2 different sides of the field. So kind of like separate experiments. And then you can see I only have 4 treatments along the bottom because these are all the soil amendment treatments relative to the weed-free control. And on the y-axis, we just have the total weed biomass, again, relative to the weed-free control, which essentially had 0 weed biomass. But for corn, you can see we had our highest weed biomass in our rye hay amended plot and our lowest weed biomass in our sawdust amended plot. And so this sawdust amendment, remember, it didn't have the most total carbon added to the soil, but it had the highest C to N ratio. So that kind of indicates to us that the C to N ratio is more important for nitrogen immobilization and reducing plant growth or weed growth. And for soybeans, we see the exact same trend. Rye hay having the highest total weed biomass and sawdust having the lowest weed biomass. So we're seeing a reduction in total weed growth, but we were also curious if this was changing the community composition. And so we looked at this a couple different ways. The first way I'm going to show you is we looked up several weed species traits in the literature. We looked up seed mass, the Ellenberg nitrogen index, which is an indicator of how responsive that weed species is, or that plant species is to nitrogen in the soil, and then the leaf area index. So we looked those traits up, and then we multiplied them by our weed count data in our actual experiment. And then we found the mean value of those 3 traits per plot. And because we wanted it to be weighted, we then divided it by the total number of weeds that we had in that plot. So again, so keeping that equation in mind, 2 of the plant traits were significantly affected by our soil amendment treatment, the Ellenberg nitrogen index and the specific leaf area. And if we look at Ellenberg nitrogen index, we see our highest index in our rye hay plots, which makes sense because we're adding a significant amount of nitrogen to that plot. And our sawdust amended plots had the lowest Ellenberg nitrogen index species on average. And then for specific leaf area, during post hoc analyses, it didn't actually have significant differences between the treatments, but you see the same trend with rye hay having the highest specific leaf area and sawdust having the lowest specific leaf area. We also looked at our weed species community using an indicator species analysis. And what an indicator species analysis does is it looks at two traits. It looks at the positive predictive value of a species and the fidelity of a species. So the positive predictive value is asking the question, does this species occur in sites belonging to this group only? So you can see in my little example that we have red species that are only found in teal plots. So that species is going to have a high predictive value. And fidelity asks the question, how many sites in this group have this species? So here we have red species that are found in every single teal plot. So this red species is going to have high fidelity. So species that have high positive predictive value and high fidelity can be indicator species of a specific treatment. So we ran this on all of the different weed species that we saw in our experiment. And we found several weed species that had a significant predictive value. And what I thought was kind of interesting about this data, what I want to point out to you, is that all of the species were pretty much predictive of soil amendment treatments that included rye hay. So that, again, is adding a fair amount of nitrogen to our soil. So I think, considering both the weed species community traits that we looked at and the indicator species analysis, that actually in the 2 years of our experiment, we're really selecting only for species that have that grow well in high nitrogen soils, because we only repeated this for 2 years. And so we may have only had time within those 2 years to select for relatively fast growing species. But if we continued this experiment for more than 2 years, possibly we would have started to see selection for species that had that would have been done better in low nitrogen soils. So we didn't see any indicator species in our sawdust amended plots, for example. But we may have started to see those if we continued the experiment. So again, going back to my hypotheses, we've looked at nitrogen immobilization. We've looked at changes in crop weed competition, especially of nitrogen responsive weeds. I now want to talk about carbon amendments and how they can change microbial community structure and function. But before I start talking about microbes, I want to check and see if there are any questions about plants, because we're going to a little bit leave plants. No? Okay. So we know that microbial communities are really important for ecological functioning. They affect both soil processes as well as plant growth. And we would expect that microbial communities would change similarly to plant communities based on resource availability. We know a little bit less about, or a lot less, I'd say, about microbes in general. But in high nitrogen soils, we would expect to see higher bacterial to fungal ratios because bacteria have generally a lower C to N ratio requirement. And we would expect to see maybe copiotrophic, R-selected, or faster growing microbes. And then in high carbon amended soils, we would expect maybe to see lower bacterial to fungal ratios because fungi have a higher C to N ratio requirement, and potentially with the ability to decompose complex organic matter. And so we wanted to look at the microbial communities in our own soils. And we did this using culture independent methods. So we didn't grow these microbes in the lab. We just were able to look at the microbes that had grown in our soils during our experiment. So I'm going to go through some data from all 3 of my experiments. And for all of them, to look at the microbial community, we extracted the DNA directly from the soil. And then we amplified different regions of that DNA to look at bacterial versus fungal communities. So for bacteria, we're going to look at the 16S region. And then for fungi, we're going to look at the ITS region. And then we use those sequence regions to come up with a taxonomic classification. And so in microbial ecology, we don't call these species, bacterial and fungal species, because we often don't know them down to species level. We call them operational taxonomic units because we may only know them down to class or order or phylum level. But I'm going to kind of use both words so that those of you who don't usually think about operational taxonomic units, that you'll understand that it's kind of analogous to species. And then when we get this data back, it's highly dimensional. So we have one soil and it will have thousands of bacteria or fungi in that soil. And it's hard to find trends looking at this data. So what we do is we create a distance matrix with that highly dimensional data. And so distances that are closer together, so smaller distances, have microbial communities that are more similar to one another. And distances that are further apart, so a higher number, are going to have microbial communities that are more different from one another. For those of you who use distance matrices, we used a Bray-Curtis distance matrix because it does well with zeros. But there are many different ways to calculate how similar or different a microbial community would be to one another. And then we plot that distance matrix in multidimensional space. And it can be difficult to see trends in an environment like that. So we use a principal coordinates analysis to simplify this data. So basically what a principal coordinates analysis does is it finds two planes within that data set that explain the greatest amount of variability of all of those points. So the X and Y axis that you're going to see in the next few graphs, they don't inherently mean anything. They're just two axes that explain as much variability as we can. And the x-axis always explains the most amount of variability. So when you see my graphs, it will indicate how much variability it's explaining. And then the y-axis always explains the second most amount of variability. And then what we do is we just color or shape the points to visually see trends based on our soil or different treatments. So, and again, we're just looking for visual clusters, points that are more similar or closer together in terms of their microbial community, and points that are further apart are more different. Okay, so we're first going to go through bacterial communities. And from chapter 2, remember this is the experiment where I buried pots in a field. And I had 8 different weed species growing. So my different weed species are going to be different shapes. And then the soil amendment treatments are going to be carbon amended, are going to be orange, unamended, teal. And you can see pretty clear separation of my soil amendment or my bacterial communities based on soil amendment treatments. And our statistics back up that trend. Oh yeah, good. Okay, for chapter 3 is our greenhouse experiment. And we see the same thing where we have pretty clear separation based on soil amendment treatments. And again, supported by our statistics. And for chapter 4, that's our field experiment. And again, these are just bacterial communities. You can actually see this is showing fungi. I think I skipped a few. Okay, well, spoiler alert, both bacteria and fungi responded really strongly to soil amendment treatments. You can see it in all 3 of my experiments. Yeah, so you've already seen these graphs. I guess just very quickly. The one thing I want to mention about chapter 4, so this is my field experiment, we're actually seeing stronger selection based on year, but within the year, we're also seeing selection based on these soil amendment treatments, which are these different colors here. Okay, so we're seeing the same selection for both bacteria and fungi. But it's not telling us which microbes are causing that change. It's just saying that they're different based on soil amendment treatment. So there are many different ways that we can go about figuring out which species are most important for causing those changes. And the one I used for this chapter 2 data, again, this field pot experiment, is the random forest method. And this is a fairly commonly used machine learning algorithm where we have the data set where we have all of our different species. In this case, I'm just going to show you fungi because there was a really strong effect of our fungal species. So it's nice to show as an example. So it takes all of our fungal species that we found in the soil. It takes a random subset of those fungi. It makes a decision tree of whether the soils were amended with carbon or were unamended. And then it does that again. It does that like 500 times. And then I take then it rates the species that were most accurately able to split the soils in carbon amended or unamended, and it gives those a highest score. So I took my 20 highest, most predictive fungal species, and I just looked at those. So I didn't have to look at the other, thousands of fungi. I wanted to see if these fungi were more abundant in carbon-abundant soils or whether they were more abundant in unamended soils. So I made a visual tool called a heat map. And what a heat map is, it's just a grid. And then each row is going to be a fungal OTU, or fungal species. And remember, I'm just looking at the top 20 most predictive fungal species. And then each column is a pot. So I had 80 different pots in this experiment. So I'll have 80 columns. And then the color, each box is going to be colored in. And each color shows microbial abundance. If the microbe was not abundant, it's going to be yellow. But if it was highly abundant in that pot, it's going to be purple. And then just for ease of analysis, I'm going to have unamended pots on one side and carbon amended on the other. And so hopefully what you can see now is that all of the 20 most predictive species were more abundant in my carbon amended soils and often absent actually in my unamended soils. So I can take a look at what these species are, but I think this table kind of highlights why it's a little bit difficult to analyze microbial data. We know these are really important microbes or fungi for my experiment, but we don't know much about them. Some we don't even know to phylum level. But a lot of them you can see are Basidiomycota, and this one Ascomycota as well. We know it decomposes carbon compounds. So we could, wood compounds. So we could think that with sawdust amendments, we're selecting for fungal species that are able to decompose wood, which kind of makes sense, but it's cool to see that like the data is backing that up. Okay, I've got a couple more minutes. I think I can run through the last thing I wanted to show you. So we also wanted to look at microbial community function, and this can be, there are different ways that people can do this, but one way people use is through shotgun metagenomics, where they amplify all the DNA in the soil and then look and see what genetic functions does that soil contain. That is quite costly, and we did not have the funds or the time to do that because all of my experiments had a lot of replicates and a lot of different treatments. So we can instead actually infer that same data using the amplicon sequences that I already had. And we can only currently do this for bacterial species. And the tool that this is called is PyCrest2, and it's just a computational tool. So I used this tool for the bacterial communities in my greenhouse experiment. But I really want to emphasize that this tool only comes up with potential estimated functions of our microbial community. It's not something that we measure directly. Basically, it takes the bacterial OTU data, it aligns them to reference sequences, places those in a reference tree, it infers gene copy number, predicts, again, this is a prediction of gene family abundance, and then infers sample pathway abundance. But this reference trees are often based on microbes that we're able to grow in the lab, so culture dependent methods. And so we're really reducing the amount of genetic information that is contained in our soils. And each step is an estimate. So we're really compounding the degrees of variability in each of these steps. So I just really want to emphasize that. I'm going to show you some results, but they're estimates. But what we did see is that our potential metabolic pathways that were more abundant in unamended soils compared to carbon amended soils in this greenhouse experiment were generally synthesis pathways. So right now, I only am showing you the pathways that were more abundant in unamended soils. So the microbes are doing secondary metabolite biosynthesis, nucleotide biosynthesis, carbohydrate biosynthesis. And this is kind of in contrast to the metabolic pathway estimates that were more abundant in carbon amended soils. So these metabolic pathways are generally more degradation pathways. They're secondary metabolite degradation, amine degradation, alcohol degradation. And actually this unclassified super pathway at the top, that's just all the pathways that we don't know anything about. So I think that highlights some of the variability that and why it's difficult to analyze microbiome data. But I think that kind of shows that microbial communities can really be selected for based on nutrient availability, and then they can have potentially different functions. And that has ramifications for things such as carbon use efficiency we're in high carbon soils, we may be just degrading those carbon compounds and have that may explain why we see lower carbon use efficiency in carbon amended soils. And then that may have ramifications for soil carbon storage. But of course, my research did not address these questions, but I hope that it can highlight the importance of the microbial community for ecological functioning. So we've addressed all three of these hypotheses, but I want to zoom back out and say that nitrogen immobilization is really important for weed control. People use cover cropping as a weed control tool and mulching as a weed control tool. And both of these are probably immobilizing nitrogen. There's been several studies, especially within cover cropping, that have looked at nitrogen immobilization. So I hope that this research can go improve cover crop design and mulch selection and use. And I also hope that we can increase the use of nitrogen immobilization in the future as our ag systems become more data driven. So we might be able to go into a field and identify areas where we have nitrogen responsive weeds and high soil nitrogen availability, and then think about use our increasingly accurate soil carbon decomposition models to figure out where we should put that carbon and which source of carbon is going to be best for mobilizing nitrogen during the critical period of weed control. And then take an applicator and actually apply that carbon in specific areas in the field. So yeah, I want to make sure I leave time for questions, but also I really want to thank everyone who made this research possible. Of course, my committee and everyone who's come into come in and out of the labs that I've been part of, as well as people who've helped sponsor my work, been mentors to me, and helped with statistics and field work. And I also want to just do a brief shout out to all the people who've made my life outside of Cornell really meaningful and made my time here just really wonderful. So I think I have a couple minutes for questions. I'll leave it on this slide. Okay, Marvin.
So if you use a cover crop for nitrogen immobilization and then you stop, you get a big flush of nitrogen release, or are you sort of forced into a situation where you have to keep adding carbon for a long time to keep your weeds down?
That's a great question. Oh, yeah, so Marvin is asking about, we're going to say we're using nitrogen immobilization in a cover crop, and then we are no longer using a cover crop. Are we just going to have a flush of nitrogen? And I think the short answer is probably yes, but that it's going to come at a slower rate. And depending upon the cover crop that we're adding, especially if we choose one with a higher C to N ratio, we're going to not be adding a lot of nitrogen. So it'll just be the nitrogen that's already in the soil, and then that will then become available. So I think that kind of plays into figuring out how long we want nitrogen immobilization to be happening, and what crops we're planting. Like, we probably want that nitrogen for some crops, and others we don't need it. So yeah, so that would definitely play a role. Time definitely plays a huge role. In the back.
Sorry if I didn't catch this, but
No.
we were looking at the immobilization in the first study.
Yeah.
And you were measuring the nitrate and the resin. Were you also looking at ammonium?
Yeah.
I'm sorry if I didn't.
No, that's a great question. So he was asking about nitrate and ammonium. And so for that particular experiment, we only looked at nitrate because we were just using resin beads that would absorb anions. However, you can get beads or resins that absorb anions and cations. And actually in our field experiment, we did look at nitrate and ammonium. And we saw reductions over the season in nitrate. But actually, interestingly, we saw a slight increase in ammonium levels in carbon amended soils. Yeah, so that's a great question.
I was wondering if it could be shifting forms.
Yeah, I think, yeah.
Some must come out of immobilization also.
Yeah, exactly. So yeah, it's not like we just have, well, yeah, this is great. So we don't just have nitrogen just standing there. It's always in flux. And so exactly like you said, we think that we're actually seeing an accumulation of ammonium in some of those soils because it's just not transforming into nitrate. So it's, differentially abundant for different plants, which plays into the selection for different plant species. Yeah, thanks for that question. Oh, okay. Oh, Aaliyah has a question.
So on around in the mid 40s, probably around 545, you said how it showed how these the communities they spatially separated not only within treatment, but they seem to spatially separate even more by year. So like in 2020, they clustered more to so together than 2021. No, yeah, right there. 2021, they really tend to separate. And do you think that's potentially due to a legacy effect of, and do you think that separation would expand even more if you continued it over the number of years? And I guess the final part, sorry for the long question, is do you think if you expanded this, since microbial communities might be more quickly responsive, that if this was perhaps like a 5-year study or a 10-year study, you would see a similar trend in terms of separation in weed communities and potentially separating based off of fecundity or RK selection?
Oh yeah, okay, that's great. That's a great question. So Aaliyah's asking about separation between years, and then also how we're seeing like a greater spread here in 2021 compared to 2020. And there's so many parts of that, and I feel like I'm kind of going to go off on a bit of a tangent that's maybe even unrelated to what you just asked. So one thing that we noticed in this experiment especially is that we're actually seeing really strong, I didn't mention this because I didn't think I had enough time, but so both of these points up here, they're the rye hay amended treatments and the sawdust and rye hay amended treatments. And all of these points down here don't include rye hay. And so we know that a rye hay actually has a lot of allelopathic compounds. And so we're thinking that potentially here, we're actually seeing selection based on those allelopathic compounds, or they also are adding, rye hay is adding a fair amount of nitrogen. So it could be a nitrogen response in the microbial community. It could also be, I think because these are so tightly like kind of overlapped, I almost have the idea that it's an allelopathic compound response or something specific to rye hay itself. So I talk about that in the paper a little bit, but we don't, usually people use allelopathic compounds as a weed control tool, but actually these two treatments had a fair amount of weed biomass. So we think that the allelopathic compound is selecting for microbial communities, but it's not seeming to select for plant communities. What seems to be the stronger selector for plant communities is the amount of nitrogen. And we are seeing already a little bit of selection for nitrogen responsive plant species. Yeah, so does that kind of answer the different parts?
Those nitrophilous weeds tend to be particularly fecund. They tend to be large seed producers on top.
Yeah, we didn't look at seed numbers. That would have been a good one to look up or count, but we did not have time. But we did look at seed mass, and we really thought seed mass might, because usually fecund plants will have smaller seed mass, but we didn't actually see a response in seed mass yet. But maybe if we continued for a longer period of time, we would have. I don't know.
So I've got one from Geneva, Caroline Marshall said, can you tell us more about 3 fungal species that were present in both treated and untreated plots or bacterial? The heat map slide, all yellow except for the 3 species.
Oh yeah, okay. Thanks, Carrie. Okay, yeah. So Carrie is asking about these 3 species that are in our unamended plots, which are the only species that are in unamended plots that were most predictive for carbon amended versus unamended. These actually, these 2 species here are still more abundant in unamended soils than they are in carbon amended soils. You can see they're kind of like blue and light green in our unamended soils, but they're actually purple in our carbon amended soils. So that means they're more abundant. So I still think that we're selecting for increased growth in carbon amended plots for these 2, but this one down here is actually more abundant in unamended soils than it is in carbon amended soils. So if we go to this next slide, we can see it's an ascomycota, and we can see, we know it down to species level, but Botryticum, I didn't see anything in the literature that would like particularly say whether, I didn't know what conclusion to draw from it. It's, you know, we can see this fungal species, it can cause diseases, but it didn't seem particularly, I don't know what else to say about it. Yeah, so yeah, I think this kind of highlights some of the difficulties in analyzing microbial communities, is that we don't always know what the species are, who they are, what they do, what the importance is. Sorry, Carrie.
Any more questions? Okay, well, thank you very, very much.
All right, cool. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.